Welcome again to the 8th webinar of the online edition of the Protein Electrostatic Conference. So if you see our program, we are until the beginning of April, and soon we'll have a few more talks arriving until the end of April. I would like to give to tell you that next week, and only for next week, the talk will be shifted on Tuesday, the same time, and so there will be another flash talk from a PhD student, Manuel Ruiz, and then the full talk will be from Anna Damjanovic, but only for the next week. So the speaker of today is Tom Laue. Tom Laue is Emeritus Carpenter Professor of Molecular, Cellular, and Biomedical Sciences and Professor of Material Sciences at the University of New Hampshire. He is the founder of the Biomolecular Interaction Technologies Center. He received his bachelor degree in natural sciences from Johns Hopkins and his PhD in biophysics and biochemistry from the University of Connecticut in 1981-80. And his postdoctoral studies were done in the University of Oklahoma, and he also worked in the deep space program of NASA. So today he will be talking about measured immunoglobulin and monovalent anion binding. Thank you, Tom, for being here.
Thank you for the opportunity to speak. And let me get myself up and going here.
Yes, we can see your presentation.
Okay, good. All right. Just want to get myself a pointer. Good. All right. So I'd like to talk about measuring charge on proteins and how I think measuring the charge on proteins will make some of your modeling more accurate. And protein charge is important both from a very fundamental aspect in terms of how what's called the quinary structure of cells develops, how you can have these high concentration complex mixtures of molecules, not have them all come crashing out, and how they can still find one another and bind specifically. It has very practical consequences as well with respect to the development and formulation of biologics for biopharmaceuticals. And I'm going to show you today that we've actually known how to measure the charge properly for 80 years, and that people have known about anion binding to proteins for over 70 years. And somehow a myth has developed that needs to be changed. So I'm going to talk a little bit about what is protein charge, what are the various measures of protein charge in solution, the myth that has developed about being able to measure the charge, and the facts about being able to measure charge. It's not hard to do, and you get a hard and fast number that's accurate. So I'm going to talk a little bit about how you measure charge, demonstrating the accuracy. That's actually a little harder to do than it may seem. Took about 14 years of work in my lab to do that. Then we'll turn to the system at hand, which is immunoglobulin G, the antibodies, and look at the charge on antibodies, fragments of antibodies in different solvents, and see what that tells us about the calculations of charge versus the measurement of charge. And then what do we know about ion binding, anion binding in particular, and why the current calculations that are done are inaccurate, and what can be done to improve the accuracy. So what's the protein?
Sorry, can you speak a little bit louder, please?
Oh, certainly.
Oh, thank you.
Okay. So what is protein charge? And let's start by what it is not. It is not what you map out on the surface of a protein. These are proton binding sites, but it's not just proton binding that's important. It's not a hard potential in solution. So when we talk about the interactions between adjacent molecules, you're not looking at a hard potential, but one that has a rather long distance dependence to it. Charge, of course, is a system property. It depends upon the ion concentration, depends upon the ion type. It's an average value. You can get an idea as to how broad the distribution of charge is by looking at the titration curve. The slope of the titration curve is proportional to the width of the binding curve. And it does assume, all this is going to assume an instantaneous reaction, which means we meet the ergodic hypothesis that the time average and the ensemble average are the same. Sometimes that's true, but in a solution of real proteins, you can have things like deamidations or silations, which can give you that where that no longer is true. Lots of terms that are used for charge measurement. The fundamental measurement that's made is the electrophoretic mobility, which is just the velocity of a boundary divided by the electric field that's causing the motion. You'll hear people talk about the zeta potential. That's the electrophoretic mobility, now adjusted for viscosity and for the dielectric constant D, and also for Henry's function, which takes into account the effects of the electrophoresis itself and how that impacts the electrophoretic mobility. There's the effective charge, that's the electrophoretic mobility times the ordinary translational frictional coefficient. This effective charge would be the charge that you would use if you're going to calculate the thermodynamic non-ideality due to charge repulsion between molecules. It's that one. That charge can be measured directly without having to determine the frictional coefficient or the Stokes radius from steady state electrophoresis. And I'll show you some results from that kind of measurement. It takes a long time as compared to mobility measurements, which is why it's not done routinely. Then there's the Debye-Huckel charge. This is the effective charge now, taking into account the Debye-Huckel ion cloud, removing the effects of the Debye-Huckel cloud, and again, taking into account the electrophoresis process itself in Henry's function. Henry's function is going to change the results by about 10%. So it's not as though it's a huge change. The kappa that's found in the Debye-Huckel Henry is just the inverse Debye length. So it's dependent on the square root of the temperature and the ionic strength. A is the sum of the Stokes radius for the protein and its counterion. And I'm going to use the word charge. I will actually speak in terms of valence. So it's just the charge in coulombs divided by the elementary charge on a proton. But I'll use valence just because we're used to working with valence. The numbers are far more intuitive than talking about the coulombs. So I'm going to speak about the Debye-Huckel Henry charge. Because that charge is valid if the product of the inverse Debye length and the sum of the two Stokes radii is less than value of 6. And for proteins that have radii that are less than about 13 to 15 nanometers, this is a valid description. For ones that have, for proteins that are larger than that, or conditions that are where kappa now becomes quite large, you really want to use the zeta potential. The two are related measurements, but again, the Debye-Huckel Henry charge is easier to conceive, for me anyway, is easier to conceive what we mean. The charge, the development of the analysis of charge is far more complicated than for analytical ultracentrifugation. I've worked with analytical ultracentrifuge for over 50 years, and sedimentation is a heck of a lot easier than electrophoresis. But you end up now looking at the slipping layer. This is the Stokes radius. It's the radius, this R should be the Stokes radius. That's the slipping layer out here between the protein and its bound solvent and the bulk solvent. The electric field, which stems from this charge at the center, is going to be the first derivative of the distance dependence of the zeta potential. And it's going to be a constant times the effective charge divided by the Stokes radius squared. And that's going to come up a little bit later when we look at some data from DNA. So here's the Debye-Huckel Henry as we would use it. Measure the effective charge and then take into account the Debye-Huckel Henry cloud and Henry's function, which is again a function of the product of the Stokes radius and the inverse Debye length. Now, there's been a myth that's developed over the years, and that is the charge cannot be measured. And the evidence for this comes from the fact that if you measure the electrophoretic mobility and you compare that to the electrophoretic mobility that you calculate, they don't equal one another. And in fact, invariably, the measured electrophoretic mobility is less than the mobility that you would calculate. Now, the calculation for the charge can come from titration data. Tanford did some of this. Kanan did some of this. From composition of the protein, many people have determined it from the composition using the side chain pKa's and taking into account other charged residues, the N-terminal, C-terminal, and so forth, or from the three-dimensional structure of the protein like Frank Gerd, Steve Shire have done. From these, you end up always seeing that the measured electrophoretic mobility is less than the calculated electrophoretic mobility. And a myth is developed that, in fact, there's a problem in the measurement and that there's some electrophoretic process that's causing this error. And that's simply not true. Fact is that the calculated charge is an error. And the biggest problem with the calculation is that there's an assumption that proteins only bind protons. And as you're going to see, that's not true. And it's not true to a great extent. So there's a lot of experimental evidence to the contrary. There's a lot of experimental evidence dating way back that, in fact, proteins bind anions. These are data shown here by George Scatcherd. Over here is low concentration salt, just so you know, this is 10 to the minus 2 molar. So that's 10 millimolar here. And this is 100 millimolar here. And the experiment is quite simple. You look at the isoionic pH. That is, you produce a protein in which solution that has only protein and water, and you measure the pH. That's the isoionic pH. You then add a neutral salt, and you measure the change in the pH as you add the neutral salt. Now, if nothing in the salt is binding to the protein, that isoionic point pH and the isoelectric pH should be identical, but in fact it shifts. And it shifts a bit. In fact, it shifts according to the Hofmeister series, and it shifts over concentration range in millimolar concentration range, and it can go quite wild depending upon the ion type. The other one is to look at the mobility measurement and compare it to the titration values, the expected charge based on titration values. And if you do the titration, if you extrapolate titration data to zero salt, in fact, the electrophoretic mobility data, which are shown here in these closed and open circles, and in the absence of salt, if the calculated value for electrophoretic mobility in the absence of salt is superimposed on that, you see that they align quite nicely. But if you now look at the data, electrophoretic mobility data as acquired in 100 millimolar salt, titration data, you find that they mismatch considerably. Again, Tanford and Gerd were very explicit in stating that their calculation of protein charge depended on there being no other ions present, and they were careful to reference papers that indicated that proteins do bind ions other than protons. But in fact, the assumption became accepted fact that proteins only bind protons, and that's just not right. So what I'm going to assert today and show you, I hope, is that the measured charges are accurate. These are the accurate charges. These are the ones that should be used in your models, and that the calculated charges where you only assume protons are binding are in error. So how do we measure the charge? Well, there are a number of ways. Well, we've already talked a little bit about pH titration, but the problem there is that it assumes that you have only protons binding if you're trying to calculate the charge. The other one is Donan effect. This is where you look at the difference in concentration of an ion across a dialysis membrane of some sort. And again, if you're going to calculate the charge, you have to assume that the ion that you're measuring is not somehow the other ions that are present, in your solution are also not binding to the protein. And finally, there's electrophoresis. Now, if you use these three, and this is comparing the charge calculated from serum albumin in 10 millimolar salt, 10 millimolar tris pH 1, these were conditions were chosen to match up the conditions that were used by Charlie Tanford in the 1952 paper, I believe, where he estimated the charge. And you can see that the charge that's estimated is a little bit higher than the charge that's actually measured. And we've measured the charge now by electrophoretic mobility using membrane confined electrophoresis, which is what I'm going to use for the rest of the day for a couple of reasons I'll point out. You can also get the charge from capillary electrophoresis using either constant voltage, which is not correct, or constant current, which is the correct way to operate one of those. And you can see these three all match up. You can also use electrophoretic light scattering. You can see that you get a charge that's sort of near what you predict, but the uncertainty, the precision of the measurement is pretty bad, 25% or so. It can be made better than that, but it takes a lot of work, a lot of material, and a fair amount of time. The problem here is actually very fundamental, having to do with the relative distance moved by electrophoresis and diffusion as a function of time. And so the signal to noise is terrible for electrophoresis of proteins. For larger particles, paint pigments and so forth, dynamic or chromatin. Last week, there was someone speaking about chromatin. For chromatin, dynamic light scattering would do a very nice job of giving you an accurate value of both the Stokes radius and the charge. Each of the electrophoretic methods have advantages and disadvantages. Capillary electrophoresis requires about 100 nanoliters of material, takes about 60 minutes to make a measurement, takes about 3 hours to calculate. From there, they've never incorporated the calculations in the software to ease that. So you end up having to do a lot of handwork to do the calculation. Concentration isn't terribly high. It does have severe solvent limitations. It has very high resolution. You can pick out single charge variants and get accurate estimates as to how much there is of each charge variant in a solution. It's widely available. A lot of research labs have capillary electrophoresis. The biggest disadvantage you run into is that the proteins tend to interact with the capillary walls. And when they do so, the velocities you measure are off. And you end up having to hunt solvent conditions where you can actually get the velocity and the electric field simultaneously. And so you end up losing what you want, which is what is the charge on the protein under these solvent conditions. Electrophoretic light scattering takes about 50 to 300 microliters, depending upon the protocol you use. It takes about 5 minutes to make the measurement, to make a single measurement at about 1 mg per mil. It has some solvent limitations. In particular, you have to work in fairly low salt. It has that precision problem that I pointed out earlier, but it does provide both the electrophoretic mobility, which you're going to need, and the Stokes radius, which you're also going to need to make measurements of the charge. In order to get a higher precision, you have to work at higher concentrations and extrapolate back to low concentration to get the Stokes radius. And you have to clean the cell in between each measurement. And so that's the time it takes to make a measurement. And you have to clean the cell, and that can take quite a while. The membrane confined electrophoresis takes 20 microliters, takes about 20 minutes, takes about 1 mg per mil. It can work over a wide solvent range. I'll show you some data showing that. It is accurate. It is very direct. The biggest limitation there is you can't use polycarboxylic acid solvents like succinate or citrate. It has to do with their interaction with the membranes that are used in this device. And then there's membrane confined electrophoresis. This is the gold standard for measuring the charge because the protein itself at steady state is not carrying any of the current and therefore is not impacting the electric field. It takes about 20 microliters, but it takes a long time. It takes 2,000 minutes per measurement. Again, you can work at relatively low concentrations. It has very high accuracy, but it's brutally slow. On the other hand, when you get the numbers from steady state electrophoresis, you can hang your hat on it. So electrophoresis is analogous to ultracentrifugation and analytical ultracentrifugation. We watch the rate at which a boundary moves down the cell. That is the sedimentation coefficient, gives a sedimentation coefficient. You look at the spreading of the boundary, that gives the diffusion coefficient. In sedimentation equilibrium, you run that process, you typically run the machine at much lower fields, and then what you get is finally a concentration gradient that is truly at equilibrium. This concentration gradient is a Boltzmann distribution and can be analyzed from thermodynamic first principles. The analog in electrophoresis is electrophoretic mobility, where you watch a boundary moving down the cell. This is the first derivative, the time derivative of the boundary moving down the cell. And steady state electrophoresis. This is not a full true equilibrium because there are ions flowing into and out of the cell at all times through the membranes. So it's really a steady state. On the other hand, this should be, in fact, a Boltzmann distribution, where the only unknown in that Boltzmann distribution is the charge on the protein. That's the effective charge on the protein. All right, I've made the assertion that the value that you get is accurate. That means it has real physical meaning. It's not just precise. The precision of a parameter, of course, is going to be how tightly you can pack those measurements together, or the uncertainty in a parameter when you do some modeling. Accuracy now means you're actually coming back to the physical truth of the matter. And accuracy is far more difficult to achieve, and it's very difficult to demonstrate. Now, what I'm doing and what I'm talking about today, the Debye-Huckel, the derivation of the Debye-Huckel-Henry equation, the Debye-Huckel equation is inductive. It's an inductive estimate of the activity coefficient. It has severe limitations in the model. The model itself uses a single point charge at the center of a hard non-conducting sphere, blah, 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 blah. What you end up getting back out is the ion distribution function, the Debye-Huckel cloud around the central ion. What I'm proposing is done, or should be done, is we actually turn the Debye-Huckel-Henry on its head. The ion cloud is the solvent response to the protein charge. So we can calculate the Debye-Huckel-Henry charge by simply taking the effective charge and asking, what is the charge at the center that's giving rise to this effective charge? What is the charge that's giving rise to the ion cloud around it? And calculate the charge from that, which I assert again is the correct charge. The relevant radius is the Stokes radius. The Stokes radius is developed from conservation principles, which includes the conservation of mass, conservation of both linear and rotary momentum, and also the conservation of energy. And it's that last one that's important because that means you have a conservation of charge in the development of the Stokes radius. So let's check and see if measuring the Stokes radius, if using the Stokes radius actually gives us a good estimate of the charge on a protein. And to do this, what we've done is take hen egg white lysozyme, both the wild type hen egg white lysozyme and a hen egg white lysozyme, which has had a 20,000 molecular weight single chain polyethylene glycol molecule attached to it, and then done the steady state electrophoresis measurement of the effective charge. You can see that they have very different distributions at the same electric field. The Stokes radius is more than tripled between the hen egg white lysozyme and the hen egg white lysozyme that has the polyethylene glycol attached to it. The function, which is Henry's function divided by the Debye-Huckel-Henry function, differs considerably. If you take the distribution, again, the Boltzmann, which sigma is just the charge times the electric field divided by Boltzmann constant and times temperature, you take that value and you calculate the effective charge, you can see they differ significantly. But if you go backwards, if you go through and calculate the Debye-Huckel-Henry charge, they are indistinguishable from one another, and they're quite close to the charge that's calculated. I would assert that these are probably a better estimate of what the actual charge is than the calculated charge. Another accuracy check is to see that if we make changes in the charge, now we're interested in getting away from estimating the actual charge on the protein at this point. We wanted to go back and see, well, if we made changes in the charge on a protein and measured the charge, would we get the correct slope of the line where we compare now the effective charge to the calculated charge? We're not interested in these absolute values. There's a great deal of uncertainty in the estimated charge from calculation, but are the slopes of these lines the same? And this shows that we could use either the membrane confined electrophoresis or capillary electrophoresis, and we can do either electrophoretic mobility or steady state measurements. And in fact, you end up getting back the same slope of the effective charge to the calculated charge of about 0.3. George Whitesides has done a fair amount of work along similar lines. And if you now compare these slopes, not the values of the charge, but the slopes to what you'd predict from the Huckel-Henry, it's 0.32, it's indistinguishable. Booth's equation, which is considerably more involved, gets you the same value. And if you do, Stu Allison did boundary element simulations of the electrophoresis of this protein. And you can see that again, it comes back to within the value that we actually measure. Now we were a little bit upset when we turned around. We were getting ready to publish the T4 results, and Nick Pace was kind enough to lend us some more charged mutants. These are proteins which lysine residues have been converted to glutamate residues. So the expected change in charge is 2 for each mutant. So this 2 lysine mutant, the change in charge should be 4 plus 4, because you're going from lysines to glutamates. And then this 5K mutant should be a change in charge of roughly 10. These are the calculated charges. And when we looked at the measured charges, they didn't fall on a line. The expectation is that these two would have a slope, adjacent to one another, have a slope of 1. And in fact, it has a slope closer to 1.5. But when you go back and use the pKa's that are measured, and again, Nick Pace sent some protein over to Lorenz's group. And when you use the measured pKa's, now these data fall on a line that has a slope that's indistinguishable from 1. How about for something like a polyelectrolyte? Do we get something that's correct for a polyelectrolyte? These are data for a DNA oligonucleotide, a double-stranded 20 base pair oligonucleotide. If we calculate the charge based on counterion condensation, you see that you get a value of -7. But the counterion condensation model is inappropriate here because it doesn't take into account the end effects. But Martha Olmsted in Tom Reckert's lab did a canonical Monte Carlo simulation, which includes the end effects for a double-stranded DAT. And the calculation here shows a charge of about -16, whereas the charge that we measure is about -18. And these two are within the uncertainty of the measurements and of the calculations. These calculations were made at 2 millimolar salt. These measurements were made at 100 millimolar salt, again, showing the polyelectrolyte nature of the charge on the DNA. There's something else we can do. Carrie May took a look at the charge on, and actually David Hayes some years back, took a look at the charge on single-stranded DNAs, DA20, DT20, a double-stranded DNA, and Brad Chairs lent us some quad-stranded DNA. So this is 24 bases long, but it's folded back on itself to give a 4-stranded structure. We measured the Stokes radius for these. We measured the effective charge, calculated the Huckel-Henry charge. And what we then did is take a look and see is there a field? Do we come up with a single field, a fixed field for these different polyelectrolytes? Well, the field is going to be proportional to the charge divided by the square of the Stokes radius. And by gosh and by golly, it is constant. Whereas other powers of the Stokes radius do not give you, certainly do not give you a constant value. That constant value, if we graph now the Debye-Huckel-Henry charge versus the Stokes radius squared, you get a straight line. The slope of that line is 4. I'm not quite sure why that is. It may have to do with 4 pi, but I really don't know. Anything charges up in this region would be disallowed. Polyelectrolyte theory says you can't take a small molecule and put any amount of charge you want on it. And so all the proteins, these are a whole series of small proteins that we've measured the charge on, they all fall in this allowed region. Okay, so let's go to the IgG and let's see what we learned from IgG. First of all, just as a refresher, IgGs have 2 light chains shown out here and 2 heavy chains. Each of the heavy chains is about 50,000 molecular weight. Each of the light chains is about 25,000 molecular weight. So the overall mass of this object is about 150,000. The binding, the antibodies, are going to bind out here at the CDR region at the tips of the light chain and heavy chain. Any kind of a virus or bacteria will have many different antibodies formed against it. Your body will respond with a whole host of different antibodies, each of which has a slightly different binding to bind to different epitopes or portions of the surface of the bacteria or virus that have particular structures, charge structures and three-dimensional structures. This region down here gives rise to the effector functions or the functional properties. This is responsible for binding C1q. So when antibodies bind on the surface of a bacteria, for example, they will form a hexameric ring. C1q is a hexameric protein that binds to that ring. That calls in the complement cascade, which will kill that cell. It's also responsible, this FC region is also responsible now for binding to a receptor. The FC receptor is used for removal of the antigen from the body. So the experimental system that Danlin Yang produced consists of 3 different antibodies, MAB1, 2, and 3. These 3 differ up in this tip region here, and they're all directed against different regions, different epitopes on IL-13, which is the protein up here. For each of these monoclonal antibodies, she produced 4 different subclasses, IgG1, IgG2, IgG4. These different subclasses, these 3 different subclasses, have different heavy chain regions out here. They're held together by different numbers of disulfide bonds. So there are 3 IgG paratopes. For each of the IgG paratopes, there are 4 subclasses. She also, the intact IgG in each case had PIs that range from 6.3 to 8.4. Less than 2% of any of these were in the form of dimer aggregates. There's no sialic acid on these. Those are things that you often find in the production of antibodies. She also clipped each of these, enzymatically clipped them to produce the FAB region. That's this region up here. It's going to be close to 100,000 molecular weight. So it's the FAB2 and the FC region. And she also prepared IgG from human serum. This is poly-IgG. There's all kinds of antibodies in there. She measured the charge on each of these in 10 millimolar sodium acetate pH 5. These are conditions that would be used for the development of an antibody and also for the formulation of an antibody for injection, and under physiological conditions using PBS. So what did she find? She used membrane-confined electrophoresis and electrophoretic mobility measurements. You'll see these data again a little bit later. That's what you get for human poly-IgG. We'll see that again in just a minute. But again, she made the measurements using that device. I've just highlighted for this particular monoclonal antibody and for these 4 subclasses in green, so we can get to the cut to the chase on what these data show us. You're going to find identical kinds of conclusions from each of these. First of all, if you calculate the charge in pH 5 on these IgGs, calculated charge is plus 57, plus 50, and so forth, whereas the measured charge is considerably less than that. So the calculated charge differs from the measured charge by greater than 40 for the intact IgG. It differs by roughly 25 for the FAB2, and it differs by about 18 for the FC region. It depends upon the monoclonal antibody. So it's going to depend upon, here it's plus 7, plus 10, plus 12. So it's going to depend upon the paratopes, that is, that binding region differences. It's going to depend upon the subclass. So it depends upon IgG1, IgG2, IgG4. It's consistent within subclass. So if you look at the charges that are measured, IgG4, these two differ only by substitution of a proline in for a serine. These proline mutants are used to manufacture bispecific antibodies. Here's the bottom line. You can't predict the charge, the measured charge from the calculated charge. We just don't know the rules yet. Hopefully, we will learn the rules because it'd be a heck of a lot easier to just be able to calculate them. The charge on the monoclonal antibody, on the intact monoclonal antibody, is close to what you get if you sum these two together. It should be pretty close to that. In fact, it is. But in general, the charge on the intact monoclonal antibody is a little bit less than the sum of the parts. If we go to PBS, we get a similar but slightly different picture. First of all of these antibodies, with the exception of this one, are anionic, regardless of what the calculated charge says it should be, and they're all roughly around minus 7.1 on average. The FABs, interestingly enough, the FAB2s all have a charge of near 0 with this one exception for the IgG4, IgG4 Pro, and the FCs all have, again, are all anionic no matter what the calculation said they should be. So the FAB2s having 0 charge is rather interesting. I'm not sure if there's a physiological significance to that or not. Again, the charge is not predictable from the calculated charge or from the structure of the protein and so forth. The Debye-Huckel-Henry charge is pretty close to the sum of these. It's not exactly right, but the magnitude sometimes is higher, as in this case, and sometimes lower than as it is in this case. If we look at poly-IgG, human poly-IgG, this is what you get for a monoclonal antibody. These different bands correspond to whether differences in the N-terminal blockage of the antibodies. Many of them, the N-terminal lysines are in fact cyclized so that they have no charge. There are different levels of sialic acid on antibodies, and so you usually see a distribution of charges in the isoelectric focusing. But when you take human IgG and you look at it, there's a second gel I'm not going to show you that shows you actually have isoelectric points that range from 10 to less than 4. If we now take this solution where you have this broad range of isoelectric points and you run them in the membrane confined electrophoresis device, you find that they all have a charge close to -6, plus or minus about 3. They're all anionic. If we now superimpose on top of that the charges that were measured for those monoclonal antibodies, you can see most of them fall right in that range. Some of them are a little bit more anionic than you'd expect. Some of them are less anionic. This would not be a good candidate for formulation because it has 0 charge, and charge-to-charge repulsion is important for keeping proteins apart in solution. The physiological charge distribution is narrow. Some antibodies, some of the monoclonals are outside that normal range. And here's the bottom line. Isoelectric focusing tells you nothing about the charge on your protein. Interestingly enough, if I go back, and I've done this, go back and calculate, this is the gamma globulin peak from a poor horse that's been inoculated with pneumococcus. You can see that the antibody area, this poor thing's producing antibodies like crazy, but they've isolated the antibodies, the IgGs, measured the electrophoretic mobility from which you can calculate the charge. It's identical to what we measured 80 years later. If you look at the correlation of the calculated to the measured charge, there is a fairly decent correlation between the two of them at pH 5. It's not quite as good at pH 7.4. What you're seeing though is an offset. So these values are offset by 24 in this case from the actual charge, and these are offset by about 6.3 again from the actual charge. So I hope I've convinced you that you really have to start thinking about binding of ions, monovalent ions to proteins other than just protons. What do we know about this? Well, the binding strength follows the Hofmeister series. These are data from Yacht and Gokarn showing the charge, the effective charge on hen egg white lysozyme in 15 millimolar salt at pH 5. And you can see now that in fact, this is all at constant salt concentration, but varying the ion type. Here's fluoride coming down through iodide, thiocyanate down to sulfate. And you can see that this line follows the Hofmeister series. There's less of an effect of cations. It's not too unexpected since this is a cationic protein. We have data showing that proteins do bind cations, but to a lesser extent. Even anionic proteins bind anions. This is serum albumin, looking at the charge on the serum albumin as a function of salt concentration ranging up to half molar. And you can see that over in the physiological range, the charge is quite small. This value of charge that we measure is quite consistent with some work that was done using Donan effect measurements where the chloride concentration as well as the proton concentration were measured. And you can see that the charge almost doubles as you go from about 10 millimolar salt up to 500 millimolar salt. Here's the titration data, the Donan effect data for serum albumin. And the amount of chloride bound at pH 7.4 is about -5. It's about 5 less than the charge we measure at lower ionic strengths. Song's group is now at the University of Singapore, has looked at NMR titration of proteins now, not with pH, but with respect to different salt types. And what he found for ephren, this is ephren B2 over here. And what he found is that all of the protein does bind anions. It binds them again with the strength that corresponds to the Hofmeister series. It's not the side chains. He's looking now, these signals are coming from the amide protons. And so you're looking at ion binding, in this case, anion binding to the amide protons. The side chains, in fact, do modulate the strength of binding. So the acidic side chain weakens compared to the positive, but the polar side chain, serine, for example, is quite close, and the hydrophobics are actually fairly close as well. So the side chain modulates the ion binding, but it's not where the ions are binding. So it appears to be on the amide proton. There's another model in which the anions in particular, are binding to the hydrophobic patches on the protein. I do not know of NMR data that backs that up. So why are the calculated values off? Well, the first one is we got lied to in biochemistry class. We got lied to because every biochemistry text has you calculate the charge on a protein based on the pKa's of the amino acid side chains. So there are different things that can cause changes in the charge. One of them is ion pair formation, solvent accessibility. The protein net charge is going to impact the shifts in pKa's, and proximal structural charges will also change. So these are all pH. This is all looking at proton binding. There's also territorial binding. I didn't show you the data, but in fact, factor 7A, which has 10 gamma carboxyglutamic acids in the first 20 amino acids, exhibits polyelectrolyte effects in that sequence in which the charge doesn't change that much. It binds calcium to those gamma carboxyglutamic acids. But in fact, the charge change when you bind the calcium is quite small because in the absence of calcium, other cations are going to have to be in there in order to satisfy the Bejerum length, to keep the Bejerum length proper. The biggest one, of course, is proteins bind solvent ions, and they bind anions more than cations. The binding constants are in the millimolar concentration range. This means, and this is where most of you come in, this means the initial conditions that you use for most molecular dynamics and Brownian dynamic simulations are inaccurate because you assume in your initial conditions that the neutrality is based only on proton binding. If you had a measured charge, you could make sure your box that you're using for your simulations is truly neutral, and it's truly neutral in the correct way. The protein binding, the proton binding to the proteins is relatively is not affected. So the pKa's and so forth are quite correct that are being determined. It's just that when you bring a proton onto the protein for roughly every, this is work that again Danlin did, for every 1.7 protons that you bind a chloride. Don't necessarily bind it at the same site on the protein, but it's a neutral binding event. So concluding, an accurate understanding of protein solutions is going to require an understanding of protein charge. Charge is an easily measured property. You can make, you saw Danlin did a tremendous number of charge determinations. Those are probably done in the space of a month. Charge must be measured. You can't calculate the charge. We don't know the rules yet. Currently, there are no models that calculate the charge accurately. We've got to learn the binding rules. We need to have a mix of experimentalists and modelers working together to determine what those rules are so that you can have accurate calculations. And this is work that was done in my lab over a period of over 34 years. And these are people who are either in my lab or David Afanis, Bill Harrington, were people who egged me on for doing this work. I thank you for your attention. I'll be glad to answer any questions.
Thank you, Tom. Thank you very much for your presentation. Let me start with a couple of questions, actually. So, one question is, so you can measure the integral charge, the net charge of your system. So there is no way to have some idea of where these charges are binding.
Well, the protons are going to bind where the protons bind.
Yeah.
There's no problem with that. In terms of the chloride, no. You could probably go back to Song's work, but there should be a significantly greater amount of work done of that sort. Or we can come up with what the rules are. You're going to have an exposed amide proton. You're going to have, if you have a charge nearby, positive charge nearby, you're going to repel. If you have a negative charge, positive charge, you're going to attract anions. Positive charge will repel them. That presumably would go just by the...
I see. And for example, in the case of immunoglobulins, would it be possible to have an idea what happens in the most relevant region of immunoglobulins, that is where they bind their respective epitope, by studying the difference in charge from the bound state and the unbound state?
It would be really interesting to do that. In fact, we got started on doing exactly that. We were going to look at a set of an antibody that bound, I think it was lysozyme. It was a protein that we'd characterized. And we set out to measure that. And the first thing we want to do is to measure the charge on the two components, the antibody and the lysozyme. Well, we had the lysozyme charge. We put the antibody in the system, and it didn't move, and it should have moved. I mean, it should have moved. And that set us off on about a 10-year adventure, which thankfully Steve Shire at Genentech and some other people in industry realized this was important for formulating antibodies. And it turns out that antibodies have this very peculiar capability of essentially buffering the charge. You can change the pH over a very wide range, and there's a very small change in charge, relatively small change in charge. And this probably does have physiological relevance because antibodies are going to have to function under a whole wide range of conditions. At the surface of a wound, for example, crossing the placenta and so forth. So yes, it would be a great set of measurements. It would be wonderful. I encourage anyone who wants to make them to make them. Okay. They're not hard to make. The measurements themselves, you in this course of a day, you can answer questions very readily.
Okay. Okay. Thank you very much. So I see a question from Guido Raus.
So nice talk. I couldn't follow all the details. It seems to me that, I mean, talking about binding, I mean, to me as a chemist, or it, I mean, it suggests the formation of a stable structure, so with little mobility of the bound. So that's not what you mean, of course.
It's both. So you can have what's called site binding, and site binding is what you're describing, where you actually have, you can recognize, you would see it in a crystal structure, you could see it in NMR, you would see this thing. On the other hand, there's what's called territorial binding. So polyelectrolytes territorially bound counterions. I didn't point it out because I didn't want to. There was a model showing a double-stranded DNA, and there was what looked like a squiggly line. That was 1 nanosecond of the trajectory of a sodium ion on the surface of that DNA double strand. So territorial binding occurs as well. In that case, it's not exactly what you're describing. It's
right. So because the first type of binding, as you said, should be observed also in crystal structures.
Territorial binding will not be. It's not readily distinguished in crystal structures in particular, because it, on a time average, it just washes out. It could be observed in NMR. You'd be looking for the signals. You have to be interested in finding those signals.
Right.
So in fact, a lot of the work I've done and work that I've been writing up these days has to do with, if you make a mixture of molecules, I'm interested in what's going on in time in cytosol and in serum. If you make a mixture of molecules, what's going to happen is the central molecule is going to have in its vicinity those other proteins, those other molecules with which it has the lowest free energy of interaction. And it's going to exclude those from its nearest neighbors that it has the highest free energy. So you end up with a structure to the solution in which there really is no binding as you would recognize it. But in fact, it's a structure nonetheless, and it could be important physiologically.
Thank you.
Thank you. I see Marilyn wants to ask something.
Yeah, that's really great. One question I have for maybe the group of us is, how much of this is stuff that we're implicitly including but haven't analyzed it, and how much of it are we missing? So if you're using explicit solvent and explicit ions, and if you say look at the time dependence of the ions around the molecule, and added that in, I mean, would that look a little bit like what Tom is showing us? And in continuum, I'm trying to think about how my D by Huckel, my Boltzmann and Poisson-Boltzmann, which absolutely includes implicit ions that are in the dibihuckyl potential. So, but again, I don't count those when I count the charge. So I think, maybe one thing to do before.
Yeah, Marilyn, what you would get from that would be the effective charge in the absence of any ion binding by the protein. You would get the dibihuckyl, and you should get the effective charge. Now, for small, what I didn't point out, I was trying to cram 30 pages of work into 30 minutes or 40 minutes. The amount of correction that's necessary for a protein goes up roughly as the square of the radius, that is with the surface area, which makes sense because you'd expect that there'd be
more charge.
Greater number, and a greater number of amide protons accessible to solvent as the surface area increases.
I mean, we've done Monte Carlo with explicit ions on the surface of human serum albumin, and it works very well. I mean, you can watch, your ions that have very low probability, but some to real probability.
Okay.
And I absolutely believe that. But I just, I'm kind of think we're even though we may count the charge just as protons, we're not calculating the charge without having solution ions involved in it. And that being involved in both MD and in continuum.
So it would be the.
I don't know if we're doing it right, but.
I don't know either. Well, in fact, the ions that I'm talking about are actually, in this case, site-bound. They are bound to the amide proton. So I don't think you would see that in your models unless you were looking for binding to the amide protons. But you, again, what you don't understand, you need to find an experimentalist who can make these measurements for you. It's not hard. I mean, again, 20 minutes, you have the answer, you have the charge, and it's a charge you can hang your hat on. You then go back and ask, does the charge that I calculate, the effective charge or the dibihuckyl Henry charge, does that effective charge now match up with the experimental value? If it doesn't, there's a problem with your model. It's not a problem with the measurement. That's the point that I want to hammer home here.
I mean, it sounds like there's already a lot of data that exists for some of the molecules that we're using as benchmarks. So I don't think we even need to do this. I think the question is more how to, deconstruct, how to add back the ion cloud in some way, or the ion, that with low probability binding to specific sites. So how to add that back in to correct the charge.
Okay, suppose we flip it around though. Suppose you take a protein where the charge has been measured and you set your initial conditions to say, okay, this protein has this many protons bound to it. And so I have to make the box electrically neutral. Here's what I would get if I had just the protons bound to it. Here's how many chlorides I have to bind to it. I'll just stick those on the surface and see what I get.
Yeah, I think that's...
And that, I don't think that would be quite... I'm speaking to someone who doesn't do what you do. I don't think setting up those initial conditions would be quite as hard as trying to come at it from an inductive manner like you're trying to do, which is try to look for the ion binding. I would just go backwards first. Then we could get the rules. Then you could go forward like you want to do.
It's a challenge. That's all. I'm just thinking out the challenge.
Oh, yeah.
If we can treat them as if we can consider this charge as a specifically absorbed, we can even in continuum description, we can treat them as part of a solute.
Yeah, we do that routinely.
Yeah,
I can assure you that if you shift the charge from an electrophoresis standpoint, you could put those charges anywhere and it won't make a difference. And my guess is that you're looking at an average of binding to any of a number of different sites. It's weak ion binding. That's the biggest problem challenge you're facing is you're talking 10 millimolar. It's right in the range where they're formulating antibodies, which is why I spent the last 20 years trying to help work on that.
Okay, I have a question from Jim on the chat, who says, any idea what degree of binding to disordered regions or proteins similar to folded if based on amide groups?
The work that, no. The work that Song did was done in the presence of detergent. So that may be, you'd have to go back and read his papers, and anyone who wants the copy of what I sent there, or papers that I referenced, be glad to send those along. But the fundamental answer is, I don't know.
Okay.
It's easily measured as long as the protein's soluble and solvent you're interested in. It's again, easy measurement to make.
Thank you. Could I say thank you for that and for the talk? And I guess really what I'm asking is what you think is special about your environments around the amide bonds. That, you know, what does a protein need to do to make it that special to present it for the ion?
I don't know. Chlorides tend to interact with the protons on water. I think you guys would know a heck of a lot more about the interaction of anions with water, and the strength of that interaction tends to go again as the radius of the ion increases. They get weaker and weaker, and you find that the binding to proteins gets stronger and stronger as the binding to water gets weaker and weaker. But the interaction of chloride, it's not a hydrogen bond. I thought for a long time it was a hydrogen bond between the chloride ion and water. And it's not. I read last night that no, the mixing of the orbitals, blah, 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 blah. And it lost me, but there does seem to be a specific interaction between anions, monovalent anions, and water molecules that could be mimicked again with the amide proton. But okay. I'm not a good enough physical chemist to be able to tell you about that.
Okay, Antonio wants to ask something.
Yeah. Well, first, thank you for the beautiful presentation. I really enjoyed it. Maybe I misunderstood some of your data, but there's something puzzling me. If I understood correctly, in some cases, the total charge was actually more negative than the charge from the proton binding sites alone. I think in the IgC case, is this correct, Tom?
I think, yeah. In fact, there's a case where if you calculate the charge on a monoclonal antibody and measure the charge, the calculated charge comes to plus 17. This is a pH 6, and the actual charge is minus 3.
Yeah. So in some this sounds to me a bit strange in physical terms, isn't it? I mean, the protein gets more charged by attracting ions than it was before. I mean,
the charge doesn't increase. The magnitude of the charge always decreases.
Sorry, that's what I meant. Yeah.
Yeah, the magnitude of the charge decreases.
Okay, because that sounds a bit counterintuitive to me. I would expect some kind of compensation towards neutrality. I don't know.
Well, there is a compensation. There is a compensation towards neutrality. What's interesting is the isoelectric points that you determine in the device and what you measure match out. So it's as you swing away from the isoelectric pH, you're not charging the protein up as fast as you can, because some of the proton binding events, there's a concomitant chloride or anion binding event, not necessarily at the same site.
Yeah.
I mean, we just don't know where those sites are. It would be really interesting to know.
Yeah, because I was thinking that one of the problems with the more naive calculations may be that even the pKa's get wrong. I mean, even the computed pKa's are wrong because the effect of the ions is not taken into account, and as a result, the relation between the proton charge, so to speak, and the total charge that you would get in the end is not straightforward.
Yeah,
But
so the pKa's are going to depend upon salt concentration
yeah.
and salt type. So if you go back and look at the papers by Frank Gerd and Steve Shire and those folks, and in fact, you can go back to the work of Scatcherd and so forth. These are all things they're aware of. And they actually they say, okay, is this because we're shifting the pKa's? In Gerd's case, are we shifting the pKa's because the total net charge on the protein plus any proximal charges that are near the site that we're interested in? Can we calculate those? It's essentially doing this, and are they buried, or what's solvent accessibility? So doing the sorts of things that ProPK3 would do, and then in addition, taking into account the impact of ionic strength on the pKa's. Those are tiny adjustments on top of the charge that's calculated, and they're very tiny adjustments compared to the charge measurements you make, what you observe, what you actually measure by experiment.
Yeah. For example, in the way I do my calculations, which involve molecular dynamics and so on, when I molecular dynamics and protonation on top of that, the way I make the setup is that I consider what is the ionic strength of the solution. So that gives me one equation, and then I want to have the box neutral, which gives me a second equation, and from that I conclude.
How do you do that? How do you do that neutrality?
Yeah, that's the problem. I do the neutrality considering only the proton binding sites.
That's
so
the
my
problem.
question to you is, this would be in principle wrong, right? That's the point.
It depends upon the size of the protein. For small proteins, the good news for you guys is the small proteins, the difference is relatively small.
Oh.
So for example, for the head and neck white lysozyme, 14,000 molecular weight, it's not a big impact. When you start getting up to larger proteins and something, bigger proteins, then it becomes more and more of a problem.
Okay.
Again, what I was suggesting to Marilyn, and I suggest to you, is you take a protein, you have the charge measured on the protein under the solvent conditions where you're doing your modeling, and you use that to get your initial conditions. You got the proton part right. Now you're just going to have to add in chloride or whatever you're using as your small ion in order to actually bring the charge on your protein to the correct charge.
Mm-hmm.
Does that make sense?
Yeah.
Good.
Yeah, that would be interesting to do.
Yeah, it's not. Again, I make it sound simple. I don't do what you do, so I can repair a transmission on a car. Sure, it's simple.
Exactly.
Until I actually get in there and take out the carburetor instead.
I think one of the problems we have in the modeling part is, for instance, in MD simulations, whatever, I mean, okay, the amount of ions in the box is probably wrong, but in spite of that, we don't see so many ions binding to the proteins. So I always think that maybe the parameterization of the ions is completely wrong. I don't know. I never worked on the subject of parameterization of ions, but my impression is that they are not that good, because I don't see them sticking to the protein, and that's strange, because I don't even have the enough amount of ions. So since there is a scarcity of ions, they should probably even bind more to the protein than in normal conditions, in real conditions.
Yeah.
I think they have been parameterized in water alone, mostly.
Yeah.
And the binding is very sensitive to the van der Waals. I mean, when we do Monte Carlo with them, it's really, but the thing is, when we do Monte Carlo, I mean, we see very small fractional occupancy. So in a sense, I mean, you might see, if you just add it up within an annulus around it, you might find, oh, it's really taking a couple, but they're moving around a lot.
But you could actually get, now the point is, you could actually get an accurate value for what that should be, and then go back until you get your model. Your models actually measure that. Again, for small proteins, for tiny proteins,
Well,
that it's not going to be a big impact, but you guys are starting to look at bigger and bigger objects. You got to know too, we've looked at lipids, nucleic acids, and proteins. Only proteins do this. Nucleic acids and lipids have their own peculiarities, but nothing, and they're the biggest one being phosphatidylethanolamine is not, despite what people think, a neutral phospholipid. It's actually anionic.
Wei Yang wants to make a question.
Yeah, I just before I ask my question, I just want to follow up on the discussion you guys had. Yeah, I think Antonio, if my understanding is correct, so here actually the ion binding is not specific binding. It's really so-called the Tom mentioned this as a so-called territorial binding. In the sense, you don't have to see the specific binding, right? This is in the context of DNA or RNA. This is called the counter ion condensation layer, right? You're gonna see a layer of ion. They flow around, they diffuse around very fast, but you wouldn't see actually they stick. But still, if you count the net ion, you still see the net ion, right? That's what I mean. Right, so that's a really, I'm not answering the question whether phosphate is right or not, but I don't think actually not seeing the ion sticking to the protein is a bad sign in this context, right? Okay, so my question is this, right? Also, I want to make a comment on another topic you guys discussed, right? How to set up a simulation correctly. The way to set up a simulation correctly is not really about actually a charge. It's about the chemical potential. Right, because actually when we use a small box, right? Nobody use big box. You use a small box. You have a layer of water about 10 to 14 or 15 angstrom, depending which culture you came from. I came from charm. We use a very big layer, but still 15 angstrom layer is still pretty thin layer. And so the key part is really chemical potential. You want to make sure the chemical potential you calculate for the ions in this box is equal to the chemical potential of the same ion in the pure water with the particular ion strength. That's the correct setup. What matters is not the charge. What matters is the chemical potential, right? To see whether in this part of the box you really represent the true reality. The only reference point is chemical potential, nothing else. Okay, so right now it's my own question, right? It's it's pretty. I really enjoyed this talk. Remember you something that people did actually, including myself, when I was in a younger age, much younger age. And so I remember actually that time people were interested in the ion distribution or ion condensation around DNA, RNA, right? There's some big guy, the old guy, David Babbage. I don't know who still remember him. I remember him. And so try to look into the ion distribution around DNAs. Right, time actual calculation can be very accurate, pretty good. Back in 2004, I remember David Babbage has a paper in PNAS, right? And you can really even see the sequence-dependent ion condensation. Right, there's another paper, another thing actually remind me about actually all the work by Andy McCammon, not that old, but pretty old. Andy McCammon, also the Nathan Baker, and so on the analysis of so-called water asymmetry, right? And they showed a result which is very interesting back then. That was 2008. I think probably Marilyn was in that Golden Conference too. That was my first invited Golden Conference to give a talk, 2008, when I was assistant professor. And so the Nathan report result, right, even show when you have a neutral molecule, neutral protein, there's neutral molecule, nothing else. You don't have a charge, and you still have a chemical potential on the surface, which is positive. The reason actually you have the positive potential around protein surface or pure neutral, that's not real protein, pure neutral molecule, and all because of water asymmetry. Because your water has 2 hydrogen, 1 oxygen, right? Which means by definition, even have a pure neutral molecule in a solution, if it's soluble, and you're gonna still see quite a bit of chloride ion, more chloride ion around the protein surface than sodium ion.
That's interesting. Yeah,
right.
That's interesting.
Right, and so my question, right, the real question I should really come back to your talk. The question is not about whether we can calculate the ion distribution using simulation, for instance. The question is how to analyze. When you have a trajectory, you have ions, right? Because you are talking about actually the ions which are somehow enslaved, right, surrounding the proteins, condensed around the protein surface, right? It's very hard to tell how to cut, right? Which layer of water belong to protein, how we analyze. I think that's the key part, right? And so the cool thing, I think indeed actually the only real standard to make a comparison with the experiment. It's calculate whatever experimental measurement. It's not about charge. If you measure the diffusion property, we have to calculate diffusion property. I think that's the only fair way to make a comparison. Right.
I would argue that when you use the word accuracy, you got to be very careful to not mistaken that for precision, for reproducibility, and so forth. What I'm proposing is the charge, which is an accurately measured property, should be something that you compare your results to. You're in accuracy, you're trying to tell what nature's doing. And so what you want to do is go back and make sure that whatever your analysis does comes up with that correct value.
My comment not even about accuracy or precision. My comment is really about the model interpretation.
Right.
Your charge came from your model interpretation, the equations, and the charge we calculate is based on. Also, we need to give a definition, right? For instance, when we see the chloride ion, sodium ion, we need to make a cutoff, right? Saying which one belong to the protein, right? And so that's not a fair comparison, because actually, in order to compare, we have to go back to your model, right? We have to use our simulation. We have to project our simulation data back to your model, right? I think that's a fair comparison. This is not about the top. It's not topic of accuracy or precision. It's topic of model interpretation.
I'm not sure which model you're talking about.
It means that, for example, when you do measurement, your measurement, right? When you convert your measurement signal to the charge, you have to use a model, right? You see this is charge of the protein.
The electrophoretic mobility is electrophoretic mobility. That's a measured property. So velocity and electric field, those are your two measurements. There's no model necessary for that. So if you want to walk backwards, you can take your model, you can take your data and work through the models that you think are
proper. No, there is a model. There is model in your interpretation. Your model
in interpretation, of course, there's that, and that was what that whole business about the accuracy. I agree with you totally. It took there's 14 years of various kinds of experiments, because demonstrating accuracy is a heck of a lot harder than talking about precision. So the fact that we can get the correct charge on a DNA oligonucleotide from measurement says that it's the fact that we can change the Stokes radius threefold and get back the same charge. The fact that the charge change in charge as you make mutations, the slope of that line matches what you expect from model. Those are all, they're attempted tests at accuracy, but I would stand the accuracy of the models that are used up against the anything you'd want to come up with, and I would still come back and say the charge we're measuring is an accurate value. I can't say why. The place where I would agree with you is in saying it's the amide proton. The data for that come from the NMR experiments I showed you from Song's lab. There's precious little work of that sort. I wish there were more to test it. Your comment that the surface of the protein is quite positive because of the water around it, that's really intriguing, because that would match very well with the observation that the error in the charge is now proportional to the square of the radius, seems to go up as the square of the radius. That would fit with what you're saying, and it would have nothing to do with amide protons. So that part of the model, my model, would be out the window. But it still should be something that would be taken into account when you're doing the modeling.
Yeah, that's a paper. Yeah, there's a paper published by, I think, Andy McCammon and Nathan Baker back around 2008 or 7 in GCP on this. Anyway, I enjoy your talk. Thank you.
I enjoyed it.
Can I make just a way? If I understood you correctly, maybe if our simulations were done in the grand canonical ensemble for the ions, all problems will be solved. That's your feeling?
No, that model was for the nucleic acid. It was looking at the polyelectrolyte effect, solely at the polyelectrolyte effect.
No. Antonio asked me regarding how to set up a simulation. Right? Yes. I should. Yeah. In principle, we solve, right? But the only thing you have to make sure your your chemical reservoir had to be the reservoir of the pure water with a particular own pure salt and with the ionic strength you target on. In that case, actually, yes. In principle, yes.
Because you think that the present parameterizations of the ions should not be a big problem, is it?
No, I don't know. I'm not expert on force field. I only make a comment regarding how to what's the proper way to set up simulation.
Oh yeah, and I agree completely. I mean, because this is an open system to ions, so they should go and they should come and go as they wish, right? Yeah. Okay, thank you. Yeah.
Okay. I don't see anybody else asking questions. Let me check on the YouTube. No, okay. So I think we can thank again our speaker of today. Thank you, Tom. Very nice, very interesting presentation and discussion. And I would like just to remind everybody that next week we will be on Tuesday and not on Wednesday, just for the next week. Okay. Thanks everybody. Bye-bye.
Thank you for letting me talk.
Thank you, Walter.